So much. All right, thank you, everybody. It's a real pleasure to be here. I appreciate the invitation very much. It's not really fair to be stuck in the middle of Moses Supercharger and Jillian Lau and Korean Bay. That's not a fair sight for anyone. And clearly my nerves took hold of me because I was unsuccessful in emailing my slides to Richard 4 times in a row. But here we are, finally here. Okay, so I am here to represent the ACTG Cure TSG and hopefully give a little bit of a summary of where we are and what we have going on, and hopefully leave lots of time for discussion at the end. So my name is Katie Barr. I'm at the University of Pennsylvania. There I am a infectious disease provider, and I have a lab and do some clinical research within the ACTG. I am the vice chair of the Cure Transformational Science Group. Marina Kasky is our chair, and our most recent chair, Pablo Tebas, is also highly involved in a lot of the work that we'll talk about today. I'll admit this person, if that's okay.
No, I got it. You don't have to worry about that.
Oh, sorry about that. Okay, great. All right, so what I'm hoping to talk about is kind of our 10,000-foot view of priorities for the TSG and what we're working on. Some of the studies that are opening now, that are open now and hopefully opening soon. Some of the challenges that myself and Marina and Pablo have thought about, but really want to open that up to other challenges that are perceived by many other groups. Talk for a moment about international ATI studies because the ACTG is starting to do more of those, and then hopefully have lots of time for discussion. Okay, the way the ACTG Cure TSG has listed our priorities in recent meetings has been these 3 things. 1, characterize HIV reservoirs. 2, evaluate therapeutic interventions. 3, collaborate with our community partners. In terms of the first, we have multiple long-term cohort studies that are looking at people with HIV in many different situations. So we have early ART initiation, chronic ART initiation, people who have low-level viremia despite ART suppression, people who acquired HIV while they were on long-acting cabotegravir. We have a non-interventional ATI study that is completed, but those samples continue to sponsor much research. And then we have another cohort of early initiation of antiretrovirals that is diverse in terms of participants from both the US, South America, and Thailand. I'm going to go over for number 2 many of our therapeutic trials in a little bit more detail, but that is the bulk, I think, of what our job is to try to test some of these interventions and see what is promising. And 3, hopefully for all of these trials, all of these studies, we are collaborating with our community partners about what our research priorities should be, how we ethically and efficiently conduct these studies, how we take information from recent socio-behavioral research and inform the next round of studies. So our current interventional strategies in terms of concepts fall in a couple different groups. All of the sort of bolded things I'm going to list here, we actually have studies looking into, and the unbolded, we do not, but others within the international cure QMA. There is a lot being done in the immunotherapy world in terms of BNABs, therapeutic vaccination, both to target T cell and B cells, immunomodulatory drugs to either make the immune system work better or have virus reactivate, and then chimeric antigen T cells, so to have people's own T cells genetically modified so they can better identify virus-infected cells and clear them. There's a lot of work that continues in disrupting latency. This is this term of shock and kill, where we take latently infected cells and ask them to show their virus to the immune system. There are new drugs asking if we can change the way CD4 T cells proliferate, keep themselves going over time by blocking what we call homeostatic proliferation. There's also a concept of block and lock, where cells that have HIV in them would be locked down so they can't express HIV. We don't have any studies in that regard, but other folks do. Gene therapy and gene editing strategies, I think, hold a lot of promise for the future. Right now, one of the CAR T cell studies that we have involves mutating the CCR5 co-receptor mutation so that cells can become invulnerable to infections. That's an exciting opportunity. There's a lot going on in terms of when we use our interventions, the timing of antiretrovirals and those interventions. And there's this new concept that personally I'm very excited about, that if we use interventions at the time when people are starting antiretrovirals, that allows two new things to happen. One, we can block some of the establishment or the formation of the reservoir that happens when people return to health with antiretroviral therapy. And two, you can use drugs, interventions, antibodies when there is still some virus present, and that allows those interventions to be more immunologically active. There's also a lot of work in either people who are recently diagnosed and starting on medicines, on antiretrovirals during early infection, or have previously been diagnosed and have started on early ART and then now are suppressed on ART and would potentially be participants of new trials. And again, that sort of translates into the different study populations that some of these studies are looking at: treated early, interventions during early HIV. We want to think about people with different sex at birth, different genders, for women in pre, peri, and postmenopausal states. And we also want to really better encapsulate the geographic diversity of the pandemic, including people with different virus and clades as part of their HIV. And of course, there's combinations of all of these. So this is an overview of the various studies that we have going on right now. In green are the four trials, three at the top, one at the bottom, that are actually open right now, open. In orange are some that are just about to open, and in black are trials in development. And just to highlight conceptually the different tactics that these are sort of trying to intervene upon. Many of these studies are intervening with people who are at the time of early ART initiation or at the time they start with early antiretrovirals or antiretrovirals at whatever time during their HIV, but at the time you start antiretrovirals. And that, again, is to try to block some of the formation of the reservoir that happens at that time point. A lot of these trials are also aiming to reduce the size of the reservoir, so chipping away at the number of HIV positive cells within the body. And then a lot of our immunotherapeutic studies are really aiming to not necessarily clear those cells, but to suppress those cells or being able to have a virus rebound and return to fight again. And then finally, at the bottom, we'll talk about for a minute, we have a study that it's open and may enroll a participant in the near future, which is a destination protocol for people who participate in these trials and get to the state of post-intervention control. So again, this is the same study, or the same group of studies in a different, equally impossible to read format. But all I want to highlight here is that we have studies that both have treatment interruptions, and then we have several studies that do not have treatment interruptions. And I want to talk about those in a little bit of time, because I think it's important for us to think about what's the question we're asking with the study. Do we need a treatment interruption in order to appropriately answer that question? And for many of our studies that are looking at new agents, either to cause latency reversal or ask some of those HIV positive cells to spit out a little virus and show themselves to the immune system, or we're trying to see a change in the size of the reservoir. For those studies, we're not asking, can we lead to virus suppression? So we don't need a treatment interruption, and we can just do different assays or different other studies as our clinical endpoint. So we hope that having studies without treatment interruption that will be promising and fuel the cure agenda will also mean there's more opportunities for participation, more opportunities for a different range of studies that people can contribute to. All right, I'm just going to talk very briefly about the studies that are actually open in the network. So A5386 is one of our combination immunotherapy studies. We are looking, sorry, part of it's blocked off, but we're looking at two different interventions. So this is two broadly neutralizing antibodies and an immunotherapy agent called IL-15. The IL-15 superagonist, the job of this drug is to do two things. Hopefully, make the virus serve as a latency reversal agent, make the virus show itself to the immune system, and also activate immune cells, CD8 T cells and NK cells, to better be able to clear those infected cells. So together, the hope is we're going to be a little bit more effective than just 2 broadly neutralizing antibodies alone. And so this study is, this study is open. The target for enrollment is 46 participants. As of right now, we have 17 participants, so we have a long way to go. The study's been open for at least 1.5 years, if not longer. This is sort of, I think, a very nice example of how complicated some of these studies are, how much we ask of the participants in these studies, and therefore how challenging it is to enroll these studies, despite the fact that this is actually an approach that the scientists in the field are very excited about. So I think that's kind of the crux of the dilemma of the ACTG and many other interventional cure networks. We have things that we're very excited about scientifically. They are very burdensome for participants. There may not be enough understanding of why this is a priority or not from both sides. And this is kind of the challenge of the Cure TSG that we see moving forward. More on that in a minute. Okay, the next study, this study just opened. It's A5374. So this is also a combination study. We are looking to combine broadly neutralizing antibodies plus an exciting therapeutic vaccine plus vesitolimod, which is an immunoactive medicine that's aiming to have the virus infected cells show themselves to the immune system together. So the goal of all of these combinations together is to have virus infected cells show themselves to the immune system, have the vaccine enhance the ability of our cellular immune system to clear those cells, and then have antibodies on board to help identify those infected cells and work in combination. So there are several different groups around the world looking at these kind of kitchen sink or combination trial approaches. This is one from the ACTG. Now it's important, this is looking in individuals with early ART treatment. And so this is going to be a difficult population to identify enough participants, but hopefully if we can see a positive result here, that result will be translated to a broader range of participants. All right, this is the other trial that is, one of the other trials that is open, A5385. This is the observational post-interventional controller destination cohort. So again, for any of these studies in which a participant reaches the clinical endpoint of the study, largely 6 months after all the intervention has gone away, and they have maintained virus control, they have not met the definition of ART restart criteria, so they're still suppressed on a treatment interruption. This is a protocol that they can enter into, be followed hopefully in a nice balance for frequency that is feasible for that individual, but also safe for that individual. And then we can ask the important scientific questions about across these different studies that led to post-intervention control. What are the key determinants? What worked and what didn't work? There is a nice neuropsychological and socio-behavioral research component to this. The socio-behavioral research is being led by Corinne, and I know we have one possible participant who may become eligible in the near future, and Corinne, of course, jumped right on that and says we are ready to do all of the social behavioral research should that happen. So we're excited about the opportunity to be able to scientifically study what worked to lead to post-intervention control, to support these participants as they go through a longer period of post-intervention control, and then to find out about their experience and learn from that. Okay. This is another study that is hopefully going to enter the open soon. This is A5388. This is looking at 2 broadly neutralizing antibodies at the time of antiretroviral initiation. So again, this is asking if we give this immunotherapy, these antibodies, at the time when people are starting antiretrovirals, can we work with the antiretrovirals to chip away at that reservoir and to have a better immune response so that it may not do the entire job, but the reservoir will be smaller and the chance for post-treatment control goes up. So there are studies that have shown promise in this regard, and this is trying to add more energy behind that sort of treatment, interventions at ART initiation. And this is another study of broadly neutralizing antibodies, A5389. This is looking at antibodies at the time of treatment interruption. So it's just antibodies at a different phase of the treatment time point. And in theory, with all of these BNAB studies together, we'll be able to really help understand when is the best time to use them, which are the right antibodies in which populations. Okay, on to some of the trial challenges as I think we at the TSGs see them. So the design of trials is really a balance between what our scientific questions are and how can we most feasibly and safely perform the trial. But in order to do that, we really need to engage a whole range of people from the very beginning all the way through the end. And that includes communities, but it also includes sites and providers and potential participants. And so there's a very broad range of engagement that we need for this whole concept to work. And I think, as Jillian was asked about how can we more effectively involve CABs, I cannot emphasize more that I think that beginning with conceptual involvement and having every single step from design, from recruitment strategies, from participant support, from evaluating the outcome of the study and how we interpret that and move forward, that's really important. I also think that if we think about the group of people who are really trial experts, the community advisory board that's working on the trial, the investigators, the few sites that are participating, they may have a really different sense of what the goals of the trial are, what the burden of the trial is versus the rest of the community. And so oftentimes there's a big disconnect between a provider involved with the study and a provider not involved with the study. Community involved with the study, community not involved with the study. Investigators in the cure space, investigators outside. And so I think as someone who's inside that knowledgeable space, it's important for me and the rest of us to understand we really need to increase our communication skills, our communication effort, because there's oftentimes, in my mind, a disconnect between what I think makes sense and what people who have very similar backgrounds but not experience in that specific trial have. So that's something I've become more and more aware of recently. All right, I think one of the challenges for the ACTG group is equity and representation. It always has been. And there's many, many, many different ways that we need to do a better job with equity and representation. But I just want to mention a couple. These have been hit upon already. But in terms of sex and gender inclusion, particularly with cis women, we have really struggled with this and we continue to struggle with this. So I want to mention the first trial that I was involved with at the ACTG, A5340. This trial began in 2013. We had 2 sites in Philadelphia and in Birmingham, Alabama. We enrolled 15 participants. They were all men. We had pretty good racial and ethnic representation, but we had 0 cis women. And so that I think is a real fail part. So let's fast forward 10 years to A5386, which is this study that I mentioned before, where we have an immunotherapy plus 2 BNABs. So the goal for enrollment was 30% enrollment of women. We included many different parts of that in the design plan. So there is an enrollment plan, which encouraged sites. You can have a certain number of enrollment slots, but you have to interview or you have to screen this many women in order to get another slot. There's all these different things that we are trying to do to encourage the enrollment of women. Michael, are you involved in this trial? Oh gosh. Okay, so you know the nitty-gritty details.
An emergency call
just yesterday, or yes, exactly.
Sites, and we're trying the rock vouchers.
Yeah, we had an emergency call yesterday or the day before. We're scheduling one for the Thursday of the day after with the sites and non-vouchers and statistician, and we're trying to figure out if we can sidestep some of this stuff. I think 30% was a really bold and very ambitious target. I just don't know if we're going to be able to do it. And that's why I was asking that question earlier about the leukapheresis. Apparently that's one of the big obstacles because there are leukapheresis and it's just really challenging. So they want me to kind of talk to these sites and say, well, what about this, and what about this, you know, and then what was the women's trial that we, years ago? I think Farmer did it. Grace, yeah, you know, I mean, you can do it. And then we also were telling the ACTG, the community screening, if you mandate it, you're gonna get the women. And that's kind of what's happening here, but unfortunately, these cure trials are so different than testing antiretrovirals. It's tough. Questions.
Let me comment here and we can take that question. Is that cool? I just want to follow up on Michael's point. So he is on the front lines of trying to deal with this specific trial, but there's many issues that we fail to be able to encourage the involvement of women in this trial. I think it's so leukapheresis in particular, a lot of times because of women's slightly smaller vein sizes, it becomes challenging to be able to get the venous access that's needed. So we have some things that we can do. We have a few tricks that we can do, like using ultrasound guided venous access, which surprisingly doesn't happen in a lot of leukapheresis centers. So there's technical advantages or advances that could be used. Yes.
Possible.
Yes. Building into the trial an alternative, so a large blood draw can be used if you have a woman participant who otherwise meets all the criteria but fails because of venous access. I think we need to talk to providers because a lot of providers discourage women from being enrolled because they fear for their safety. And some of those are very valid reasons, and some of them may be, as Moses Supercharger mentioned, based on misperceptions. And so we need to be very, supportive of the real concerns, but also take the time to educate if some of those aren't. All that being said, we are trying, there's clearly a stated effort, but we need to do more of the legwork, I think, to really make it feasible for women to be enrolled at a higher level of these trials. Otherwise, we're gonna go from 0 women to 10% women, and we need to make bigger steps in that regard. Okay, is there a question? Yeah.
Thanks for your presentation, Katie. Just really quick and follow up. What steps have you all done to engage women within the ACTG? Blessings to Michael for representing the community on that trial. He is not a woman. He's not one. He can comment on his own individualized efforts to engage with communities of women. However, I've not seen him on the team when the call since I've chaired it. So can you comment a little bit on what you all are doing to engage communities of women specific to the enrollment of women in this study? I can hear the passion coming forward through your voice, but for me, in hearing it, I'm seeing a lack of passion. I hear lots of intent, but can you tell me how you plan to operationalize the passion I hear in your voice around enrolling women? That's what's key.
Well, I think you stated it very well. I do believe in this strongly, and on this trial in particular, I have not been involved in the efforts to better engage women. So you're right, there's a disconnect there, and we need to do a better job. I think that, this is not, we do have women's we have women's community groups, and we have a women's science group, but for this trial specifically, I don't know. And I think I need to do a better job of knowing that answer. I will say the things that we're doing in terms of trying to alleviate some of the barriers that we know exist. For example, at our site, we have purchased, our ACTG CTU purchased the ultrasound machine for our leukapheresis unit, and we are training the apheresis unit right now into using that. We are going back and we are screening multiple women who failed the first round because of venous access to see if they, with the ultrasound, are now eligible. We are also looking at many of the protocols that we have where we have this leukapheresis requirement, making sure there is the large blood draw as a secondary alternative. So that has been going on over the past month or so to shift leukapheresis from mandated to desired, but having that second caveat. So there are things that we recognize and we know from women's advocacy groups are key issues. We have spent time over the past couple of years, because we've heard a lot of this from groups, that issues of birth control requirements are oftentimes incredibly onerous and maybe question the autonomy of a participant because we're asking them to do things that are more than they feel are required for their own and for their own safety. So we've talked back and forth for over the past couple of years pretty extensively with DAIDS about how we can be more flexible about the types of birth control and pregnancy prevention requirements that there are for any woman who would participate. But to your point, when we looked at for example, for 5386, when there was a list of all of the women who were screened and why they failed the specific, or why they did not meet enrollment criteria, the specific reasons have just been cataloged, and I think we need to have a broader discussion with the women's advocacy group to better address those. So I think we can do that.
Shall we have you on a call for April?
I would, yes.
Thank you so much, Doctor. I have three quick questions. First one, I really loved this strategy of latent reversing whatever strategy in the beginning of the cure research. But I'm wondering really what happened to the discovery of new latent reversing agents. We haven't seen so much investment into it. The second concern is around the interleukin 15, which you are using in this kind of cure study, and we know it has a challenge of neutropenia. You call it neutropenia. So what are you doing? What are your plans in handling the challenge of neutropenia kind of and the last one was around thank you and the ACTG for because I live in Africa and I see so many sites doing research with different community advisory boards. But the vibrance and the energy of the ACTG cabs is much better, much stronger than all other cabs, which sometimes even sponsored by, I would say, the yard. But the one of ACTG treatment is really much better. What are your plans to sell this vibrancy and energy to all those other cabs?
Okay, you asked such hard three-part questions.
So real quickly, can you repeat the question for the people online, and then for the sake of time, any other presentations, which one would you like her to answer most important?
Okay, I'll try to be fast. Questions from Moses Supercharger. 1, what are the new LRAs? Where are they? We haven't heard much about them. 2, IL-15, neutropenia, other side effects, concerns about that. And 3, ACTG cabs, some of them have great energy. How are we going to spread that? I'll start with the first. Actually, there's really exciting LRA work that's going to be presented at CROI this year. So there is a whole session on, there's a theme discussion on TAT inducing regimens. There's 3 independent groups that have developed strategies that seem to be promising as a new LRA. So that, I agree with you, Moses Supercharger. We had a lot of LRA talk like 10 years ago, and then nothing new for a long time. There are some new promising ones coming, so I think that's something to be excited about. 2, IL-15 superagonist, you're right. There are several concerning side effects. Some studies have seen, but it's very heterogeneous, and so it's not clear when they happen, in what participants, in what setting. So I do think there's very strong safety controls in these studies. There's pauses whenever there's any concern about neutropenia, but I agree, that's an issue, and that's something that's really going to need to be assessed over the multiple studies that have been conducted with this agent right now. All right, I'm going to try to go fast so I don't cut into other people's time. Another equity and representation challenge has been to try to have our studies reflect the global pandemic. And so the ACTG is working to try to expand some of our cure research within Africa, within South America. So we have 3 studies, one that's ongoing and co-sponsored with other networks, and 2 that are soon to enroll that are in 4 different countries in Southern Africa, South Africa, Zimbabwe, Malawi, and Botswana. We have several studies that are open either now or going to be in South America with sites in Peru and Brazil. There's obstacles in that some of our studies use broadly neutralizing antibodies, which aren't equally active across the clades of virus around the world. There's also a lot of export-import restrictions from people who have new agents, new drugs that we're trying to test. That has been a real challenge. And then, of course, there's all the inertia about doing trials around the world in new places, but we are trying to do this better. So I just wanted to spend a moment about ATI studies. I know Corinne is going to be talking about this extensively, and Jillian was asked about this as well. For studies ending or aiming to induce a functional cure, there's real scientific value in treatment interruptions. We do not have an alternative biomarker or a surrogate endpoint at this time, and we will continue to work on this, but my personal view is I'm not particularly optimistic that we're going to find one in the very near future. My experience with the ATI studies that I've been involved with is that they are really very generally safe, but they are incredibly burdensome on the participant who chooses to participate. Therefore, it requires just an immense commitment from any participant, and that really requires sites to have an extensive communication program to really educate that individual for informed consent about what they're actually going to have to do to participate safely in that study. There needs to be participant supports throughout the study, as including partner protections, and I know Corinne is going to talk about that. One of the restrictions we have within the ACTG is a relative lack of funding for socio-behavioral research, although we recognize its importance, and so that, I think, is something we need to work to change in the future. But there is some socio-behavioral research, and we need to be able to take those results and implement them as quickly as possible. And then, as was mentioned, I think the continued research to make these trial designs better is important. In terms of at-home point-of-care viral load testing, there are groups working on this. I know at Penn, the engineers have been working very hard. This was stopped with the pandemic, but we actually had several prototypes, basically of the same COVID at-home test, looking to see if we could detect virus or not. And I think the ability to have an at-home test would be revolutionary for the experience of a participant in a treatment interruption, and we should keep pushing. It is feasible, and we should be able to do this. And this could be used across the continent of Africa. We don't need cold storage. They can be inexpensive. We could be using them everywhere. So I think that's an important thing. Scientifically, we need to push for that search for a biomarker so that we don't have to do treatment interruptions. But that's a challenging goal that we need to keep working on. I just want to mention the ATI studies that will be started or have been started in Africa. So the first one is the AMP-ATI in Africa. This is a co-sponsored trial between the VTN and the PTN and the ACTG. This is a build of the AMP prevention study. So in AMP, people were given BRCA1, and they were followed over many, many years to see if it did a good job preventing HIV. For the people who seroconverted, who acquired HIV, there's a follow-up trial to see if starting antiretrovirals very early during that trial, plus or minus getting that BRCA1, was able to induce any different immunologic changes, reservoir changes, or perhaps post-intervention control if they did a treatment interruption. So this study was conducted in Africa, based on the AMP in Africa, and in the Americas, based on the AMP in America study. So the study, this is the schema, and basically participants were asked to undergo a treatment interruption, and then we waited to see when their virus rebounded. At this point in time, from the Africa study, the study is largely complete. We still have some participants being followed on antiretrovirals. This is a small study, just 13 participants, but a really unique population because these folks had all spent years engaged with the clinical trial site through the AMP study. Then there was an extensive stakeholder engagement process over the course of multiple years. We paused a little for COVID, and then the study moved forward. We did have 2 participants who had a fairly long period of time off antiretrovirals. And so all those data and all of the lab assays and the associated research is being completed right now. But I will say there were no serious adverse events. There were no major complications. Participants felt, for the most part, well supported as far as we know now. And so I think this is a model This isn't exactly how the rest of the ATI studies will be, but this is a good example of how, with good stakeholder engagement and with good support of participants, these studies can be done. And a similar study is being done largely in Peru, but also some Brazilian sites with the AMP in America study. There's a paper that's in preparation right now to sort of describe the implementation, stakeholder engagement, and early clinical data. Hopefully that will be out soon. And then there's a lot more work going on with that. The two studies that are moving forward in the ACTG for ATIs in Africa, this is also a study that is supported by the BTN and the PTN and the ACTG. This is looking at two very promising broadly neutralizing antibodies and asks, in a clade C population in Southern Africa, can these antibodies work as well as they did in a clade B population in the United States and Western Europe, where they've previously been tested? The reason the BTN and the PTN are involved is because these are antibodies that people would like to use in a prevention concept. And so understanding if they work to prevent virus rebound in this treatment context will inform whether they could also be rolled out for prevention. So there's buy-in from all the networks. This study should be opening very soon. The next study is A5417. This is looking again at the same antibodies, but given at the time of ART initiation. So again, that exciting time when there's virus present and there's the ability to stop the formation of the reservoir. There was a study like this done out of Denmark, mostly in Western Europe. And this is the question of whether we can do the same sort of, have the same sort of effect in different virus clades, different socioeconomic contexts in the continent of Africa. So this is my last sort of slide, but in my mind, our sort of large overview of the Cure TSG goals and approaches moving forward. We really have to open and enroll studies. The COVID pandemic was tough. Our trials are complicated and they're challenging, but we really need to move our logistical framework forward and get these studies open and support the sites to be able to actually do the work. We've tried to do little things like support the sites with centralized leukopak processing, incentivize cures sites for enrolling in cure trials over other trials. But honestly, we have a lot of work to do in order to make these trials open and move forward. In terms of the studies that are ATIs, we really need to provide good support across the board for communities, for sites, for providers at those sites across the whole range. And I think that what the ACTG Cure group would like to do, we now have buy-in from the leadership to do this, is build on much of the work that the Partner Protections Working Group did to develop a toolkit to specifically address the issue of partners of ATI participants and how we can protect them. So we're going to implementing that toolkit and then build to have a more holistic view of all the things that a participant of a cure study would need and how we can support them, the sites, their providers, their partners, etc. Okay, happy to have any discussion.
I'm Frank. Repeat how much time do we have?
Take it
Questions?
7 minutes.
I think maybe it's 5 minutes.
5 minutes, okay, terrific. We'll start with Michael, if you want to queue up some questions that are coming up online. And one question, or one thing I just want to add to the discussion of leukapheresis. It's not just women who would benefit from large volume blood draws, but a lot of those middle-aged white guys are on testosterone replacement therapy or have sleep apnea and are therefore polycythemic and need it therapeutically. So given how expensive it is, you can save yourself some money. Michael, go ahead and answer some questions you may have.
The one question that we had was about how does the WHO's recent sort of reconstruction around the U equals U, where they did the red light, yellow light, green light, which sort of suggests that we can go up to 1,000 copies in a viral load. How does that affect your criteria for restarting?
So the question, just in case it wasn't heard in the room, was how does the WHO's recent sort of discussion or materials about the red light, yellow light, green light, about U equals U with various viral loads, how does that affect our viral load treatment interruption, ART restart criteria for treatment interruptions? So Corinne, are you going to talk about that? Okay, Corinne is going to address that specifically because I do think that's been built into several different components of the toolkits that she and others, or she and her group have developed. But I think the science that informed that WHO concept that under 1,000 is really not associated with onward transmission, that's part of the framework for the current treatment interruption, ART restart guidelines, with 1,000 as sort of a cutoff for a lot of them. But honestly, there's a range of criteria that are being used across multiple different groups. For almost all of the ACTG studies, our harmonized criteria use that 1,000 time point as sort of a threshold. If it's met, if it's confirmed, then that would be the time that ART is restarted. So I think they're fairly well aligned now, but there is going to be another meeting in May, I believe, in Nairobi to rediscuss API and ART restart criteria and sort of get a large sort of global view of these sorts of issues. And I think that those WHO messages will be more fully engaged with at that time.
There's a space where you're gonna have this rebound, and then the question.
Well, the study, so that those interventions where there's rebound, but then they're anticipating that the body reacts and and then it's gonna go back down eventually. So you can't, on those studies, you can't restart at 1,000, and that's problematic.
I understand what, so you have that, all right. So the fact that we know 1,000 has good science behind it can inform our education about what we do with those viral loads. So if, the informed consent process of engaging a participant in one of those studies that has allowance for a viral load over 1,000 for a period of time to see if that immune response will be able to pull it down. That period of time needs to be recognized and discussed as, okay, we all know from multiple different levels of science that when the viral load is at that level, transmission can happen, and we need to be aware of that and use all of the precautions that we had, but in particular be informed about what that means. And so during that relatively short period of time where we're monitoring people closely with detectable viremia, that has to be sort of a part of that discussion and a part of that understanding that it's a short time. We're waiting specifically to see if the virus is controlled by a newly empowered immune system. That is kind of the important part of those trials, to see what happens right then. But it's a very important time for discussing the risk of transmission.
We had another question online here. The question was, how are you ensuring that people in Africa are truly informed about the lack of benefit and the risk of pausing ART? How are you engaging with community leaders and members to be sure participants are aware of the potential health risks and the risk of partner transmission during ATIs?
That is obviously a very important question. I think that at all of the sites where we're going to do this study, that process is extremely important. Using materials, investigators, ethics, regulatory, community members from each site will be essential to have those discussions and to make sure that, given the context, the cultural context at that site, we understand what it takes to have an informed consent discussion. It may be a little bit different from place to place. There may be different issues. So I think that's an amazing question, and the 2 minutes I have right here is clearly not enough to fully address it, but that is front of mind as we're doing these studies. That's part of the stakeholder engagement process. That's part of the site support process, so that you can have a very strong conversation and say, this is an altruistic study. You're doing this to support the science without a realistic benefit to yourself, and therefore you have to understand what the risks mean, what you're undertaking in terms of all the things that you mentioned.
Thank you so much for that amazing presentation. This question is not to do with what you have been presenting around, but it's been troubling me for quite a long time. So much samples that have been taken from my continent, Africa, for research to the Western world. And I'm wondering, do you have future plan with the ACTG TST of creating an elite controllers bank from the samples that you have taken, or not, even the new ones, because of recent I've seen so much research into elite controllers, how they're going to be used into curing people of HIV. But do you have like a global bag for the samples of these elite controllers, especially from Africa? But something tells me that our elite controllers are likely to be more.
The short answer is no, we do not have any plans to do any sample banking for elite controllers. But the longer answer is that one of the things that we are engaged with is looking at people who have participated in any of these trials and these sites who have control after treatment interruption or after an engagement, after an intervention. And for those studies, we have this destination cohort, which will be available to any of the participants at African sites, at US sites, at South American sites. And from those individuals who demonstrate control, we'll be able to bank those samples. Those will largely be stored at the institution that's doing the study or in South Africa, likely from African sites. And those samples, those participant experiences, I think are going to be essential for the next steps in cure research, because elite controllers and post-treatment controllers both really inform our understanding of what do we need to do to elicit suppression of the virus. How can we get there? I would love to see control bank everywhere in the world. But that's a little out of the TSG's purview. I think that's a great idea. But for post-treatment and post-intervention controllers, I think that would be a part of some of these new studies.
Just the last one. The room, and then we'll move on.
Yeah, thanks for the wonderful presentation. So I'm just curious, and I'm not sure this is a stupid question, but I know for treatment and prevention trials, the sites are picked because of the prevalence. Yeah, it's very hard for me to say that word. I imagine. So I'm just wondering what influenced the picking of the sites for the cure trials? You know, like Africa, South America, North Europe, or yeah.
That is literally a multi-million-dollar question, not stupid at all. There's millions and millions of dollars going into that site. So for our ACTG studies, they're conducted at ACTG sites. So these are sites that have clinical capacity around the world to be able to do these studies. Now, which of these sites will participate in a cure study is sort of a second issue. Largely, we're looking for sites that say, we think we have participants who would be interested in this. And therefore, they're excited and they feel they have the capacity, they have the support, they have the engagement within their local community that they think it could be successful. And then there are some other issues. So like I said, with the broadly neutralizing antibodies, some of them have better coverage in different virus clades around the world. And so for our clade B BNAbs, we've largely done studies in the Americas, including Peru and Brazil, because that's a clade B infection largely. Some of these studies in Southern Africa right now are being chosen because we want a clade C virus population. And so there are some sites with excellent ACTG sites that have a very diverse group of virus clades in their communities, and we're not including those because we don't have enough data about whether those antibodies work against the range of viruses in that community. So that's true in places in Eastern Africa and Western Africa. Whereas in Southern Africa, we know there's a lot more dominant clade C virus. They have preclinical studies and other studies in clade C, and so we have more sort of belief that these antibodies are going to be effective in that clade, and that's why we're trying to see if they work in this context.